All right, so the materials, this is the procedure for heat fixation. All right, so we have goggles, lab coat, inoculating loop, Bunsen burner, glass slide, clothespin, matches, plastic pipette with water, and 3 plate cultures, okay? Now, the other day we used a culture in a slant. Today we're using them in the plates. So I have 1 known culture and 2 unknowns. So I'll bring this over. And this is what bacteria looks like in a plate. And you can see these small round colonies that are by themselves. That started from 1 cell and multiplied overnight to make thousands or millions of cells. So they all came from the first cell. So if you were isolating bacteria, you would look for an isolated colony. All right, so step 1, attach the clothespin to the slide. Step 2, put a couple of drops of water on the slide, probably toward the end, and you always keep it tilted downward. Open up which culture you want to use. Now in this lab, you've got 1 culture of E. coli. You know that's going to be gram-negative, and then you've got unknown B and unknown C. I know what they are, you don't, okay? So you open up the plate, and you want to keep it exposed as little as possible. Sterilize your loop before you touch the plate. Cool it off. Now, a good way to test to see if it's cooled, find some agar with no bacteria and touch it. And if it sizzles, it's too hot still. Okay? Now, if you were looking for an isolated colony, you would pick up 1 little tiny circle. In this lab, we really don't care what they are. And by the way, this culture looks like it's contaminated, but I'm going to use it. So I'm going to scrape some off with the loop. Cover the plate. Meanwhile, you know you have bacteria on this loop, so you don't want to touch anything. Spread it evenly into the water. Remember, there's millions of cells there. Before you put this on the table, you have to sterilize it. You'll see the bacteria burn off of it. Then you may put it down on the table. You don't have to worry about the surface, they're fireproof. Now you can put up your mask. Just don't want it fogged while you're using the flame, okay? So as soon as it becomes fogged, pull it down a little bit. Then you're gently going to pass the slide to the flame. Now here is where your patients are going to determine how well you heat fix your slide. This is called heat fixation. You're going to fix the bacteria permanently to the glass slide. Now glass heats up very quickly. So initially you want to keep it on there a little bit, and then you're going to start to see the sides dry. Really no set time limit. Pass it through. And then wait. Now, the more water you start with, the longer it's going to take. So a smaller drop is always better than a larger drop. Now, you may wear gloves when you start. I don't wear them. I've been doing it a while. You're welcome to wear the gloves. A suggestion is when you're starting, yes, you should wear the gloves. Now, several times throughout the lab period, you're going to wash your hands. Just randomly, in case you've touched something on the surface or the equipment. And the rule of thumb is to sing happy birthday twice while you're washing them and lathering them with soap. Normally, we would have hot water or warm water. We don't in this building, so yeah. So you can grab a little hand sanitizer if you'd like when you're done. All right, so it's beginning to dry. See the layers forming. Now, if you go too fast, you run the risk of cracking the slide, having the slide shatter. And if somebody doesn't have goggles on that's close by, they could possibly get the glass in their eye with bacteria. So it's very important. Everybody near the heat fixing stations has the proper eyewear. And an apron, but eyewear most important. Then you're going to notice it's going to start to dry very quickly as the glass gets hot. See that? Now the other thing is if you get globs of bacteria, because there's a tendency for you to grab as much as you can to make sure you have it. You put that on there in globs, it's going to turn black. And then that's going to be permanently on your slide. So the idea is to get as few bacterial cells in the water as possible so that you see them evenly spaced and stained. I've pretty much got a lot here. I should have a lot less than that. Now when you're heat fixing it, the gram-negatives will probably fix a little faster because they have more fat in the cell wall. But not necessarily. Staining will determine that. So there we go. So I'm seeing it actually just dry. And there we go. So it's fixed. Okay. Now you've always got to remember what side is which. I put the stain on that. So you see the bacteria is permanently fixed. All right, now we're ready to stain this, but you're going to time this for at least a minute. So someone time it for me, because you want to make sure this is cool, because if you add the stain right away, it's going to sizzle and burn. That's not good. It's going to be totally cooled off, so about at least a minute. Okay? And remember not to touch it. That's going to cause a blister. That's hot glass. Okay, just like you were taking Pyrex out of the oven. Same thing with the loop. It's a wire loop, so it's metal. Will get burned on them. Now I'm going to bring these stains over to the sink. Now, because I'm taping it, I'm moving everything. When you're in the classroom, the staining stations are back near those sinks in the back of the room. We never keep the stains close to the flame. Okay? I'm just doing this for demonstration purposes. All right, so the names of these stains are crystal violet, a purple stain. It's also called a primary stain. You use it first. Graham's iodine. It's a special iodine. It's brown. Different from Lugel's iodine. It's named after the scientist that discovered the staining procedure, Christian Graham, and that's the mordant. So once you put the primary stain on, we put a mordant on it to fix it to the stain. And then you have your decolorizing agent. That's going to release the first stain if it's going to be released. It's 95% ethyl alcohol. And you will add that drop by drop until it runs clear. That's probably going to be done very slowly and carefully. Then you will have a counter stain or a secondary stain. This is the primary stain, that's the secondary stain, or counter stain. And it's safranin, it's a red stain. Now let's go over the chemistry of what happens. In the cell wall of the bacteria, you have protein and fat. Gram-positive bacteria tend to have more protein called peptidoglycan. Think of it, the 3 Ps. Positive, protein, purple. They stain purple. So gram-positive bacteria with a lot of protein stain purple, because the mordant allows the stain to fix to the proteins. Okay? Gram-negatives have more fat in their cell wall. So when you add the alcohol, this is the chemistry behind it, it melts the fat. And when it does that, what's released from the cell? The primary stain. So you'll see the purple stain being released. And then you're back to clear cells. So when you restain them pink, or red. Gram-negatives appear red. Now you will learn when we get into prokaryotes that gram-negatives are usually more pathogenic or disease-causing than gram-positives. These are usually a surface bacteria and they're stronger. These are usually intestinal bacteria or bacteria that invade the body. And they usually have a capsule layer with poisons and toxins, and they can all be either bacillus or kachi or spiral. All right, and then you have bibulous paper, which you will use at the end, also called blotting paper. And that's what you blot it dry with. We never rub it dry, we blot it dry. And you rinse between each step. It's a 4-step process. So this is what we call the staining procedure. So this is the second. All right, step 1. You're going to flood this slide with crystal violet over the sink. And don't be afraid to use the stain. Now, keep in mind, if you get the stain on you, it's going to stain your skin, so you should have gloves on, especially your clothes. Rinse it, and that's why we don't use these sinks. See how they're splattering? We use the back sinks. All right, so I'll rinse it off, and it should appear purple. Okay? Then I take my iodine, flood it with iodine, rinse, and it's usually instant, at least 15 to 20 seconds maybe. And it should appear black the more it has reacted with the stain. The most important step is the decolorizing step. If you go too quickly, you can over-decolorize. If you don't do it enough, you can under-decolorize. So it's going to be drop by drop until it runs clear. So you're going to hold it over the sink, tilt it, and slowly add the stain drop by drop. Now those of you in the class, you could probably see the purple stain being released at the bottom. See it? And it'll be released on any of the bacteria. And you're going to watch that drop dripping off until it's clear. See how it's running clear?
Okay.
See that? You'll rinse. And usually the gram-negatives, you'll make a clear zone, but it'll still be kind of purple. That still looks like there might be some, so I'm going to try a little bit more. But it's running clear, so I'm going to go with it. And then the last step is to flood it with the cream. Thank you. Rinse it. And it does appear more red. Everyone see that?
Mm-hmm.
And then you take your bibulous paper, blotting paper, and then you place it down and you fold over, blot carefully. Now there's nothing like cracking the slide at the end and then you're going to start over. Okay? Do it a couple of times, use a whole piece. And then you're ready to observe this under the microscope. And it'll tell you whether it's gram-positive or gram-negative. It'll tell you the shape of the cells. Are they kachi? Are they bacillus? Are they spiral? Also, you can see if they're in chains, strepto, or in clusters, staphylo. Okay? You'll start on low and go to high. And that's the gram stain.